ಎಲ್ಲರಿಗೂ ನನ್ನ ನಮಸ್ಕಾರಗಳು. I'm extremely delighted to be here this evening with you and would like to thank our organizers for that. And to Dr. Virat for a very generous introduction. I recognize and realize that the audience is mixed and the topic is highly technical. I'll try to keep it as simple and non-technical as I can, and in that I need your good wishes. Plant breeders have been doing from very beginning two things: creating genetic variation and from among the variations selecting what they want. The tools they use: hybridization. It is the first tool that was used. It is possibly the major tool that is being used. It utilizes the variation, as Dr. Virat said, that nature gene edited for us over a very long period of time, and evolution allowed to exist in the crop species that we domesticated. Mutagenesis came along earlier that generates changes in the genes that exist in the gene pool or in the personal crop by using physical or chemical agents. Then came along GM technology, transgenic technology, that brings in new genes that create novel traits in the crops, but it's painted red in smaller letters for reasons I'll discuss later. The currently available tool is gene editing. Gene editing does everything that mutagenesis does in more controlled and in more efficient way. It can also make transgenic technology much more safer than it is today. Transgenic technology poses primarily concerns because of two basic reasons. One, that it puts in genes that are not known in that crop. Concern comes from that. Whatever genes that you put, whatever DNA sequences that you put in. The second concern arises because of the site of the genome where the gene gets integrated. Since integration is random, there is no way to decide where to put in the gene that you want to put. Random integration creates primarily two types of problem. One problem is directed at the gene that you put in. Its expression level may be affected. Its stability of expression over time may be affected, and there may be gene silencing over time. The second problem posed is towards the genes that are present inside the genome of the crop. The integration of transgene that is put in may inactivate some genes that are undesirable. They were inactive earlier, but because of this gene integration, they become active. The other thing it can do is that it can inactivate a gene that was beneficial to the plant, but because the transgene integrates within this gene, that gets inactivated. In biosafety evaluation of the risks from the genes that are put in and the other sequences that are put in, that's simpler, straightforward, and not that demanding. It is the second one that is very demanding, the effects produced due to the site of integration. Gene editing allows you to select a genome and then place your transgene precisely in that genomic site. In human genome, some safe sites have been identified, but like any evolving knowledge, what was considered safe yesterday is no longer considered that safe today. I'll give you an example. The gene CCR5 that is involved in HIV infection of human cells. It was considered to be safe, not only safe but beneficial. You inactivate by CCR5 by putting in a gene, you make the person resistant to HIV, but you make him susceptible to something like flu and West Nile virus. So, if a safe clone was found in the genome of the crop that you are working with, you can integrate transgene there, and bulk of the evaluation process then should be eased out, and only certain direct provings had to be done. I'm not going to talk about transgenics at this point of time. I'll talk towards the end very briefly, but briefly refer that it's a technology that is not looked favorably in this country, and after 2002 there has been no release for commercial cultivation. Mutagenesis, as I said, makes changes in the genes that are there. The changes are random, they are in low frequency. There is no control that the breeder can exercise on which genes to mutate and what changes that will come into it. As a matter of fact, over the last decade, the interest in mutation breeding has gone down drastically, and as you can see, there are only 94 varieties registered, included in the International Atomic Energy Database after 2010. The gene editing is a tool that mutates the gene that the experimenter decides on, at the same time induces the changes that are decided by the experimenter. And the frequency is quite high, 3 to 10% of individuals get changed, and some of them may be homozygous mutants. So gene editing provides almost complete control on the gene mutated and the type of mutational change. In simple terms, gene editing procedure involves programmable seizures, technically site-directed nucleases, that induce cuts into the DNA at the decided site, and these cuts are repaired by the cellular DNA repair machinery, and during the repair process, the genetic changes are induced. The first seizure, so to say, was created in 1994 as zinc finger nuclease. And that is when gene editing began, the modern gene editing, the third generation of it. It picked up speed when talents were produced in 2010. The reason being that redesigning of ZF and zinc finger nucleases was very extremely demanding technically. Talents still they are demanding, but they were less demanding, so picked up speed. It picked up speed, and after 2013 there was an explosion in this field. The reason was that in 2013 CRISPR-Cas9 system was described and adapted for gene editing. These two seizures have DNA binding proteins that are transcription factors, and to them are attached the domains for cleavage of the cutting of the DNA. But CRISPR is the nuclease that is inactive otherwise. It is activated by its association with an RNA called guide RNA. In this case, it is referred to as single guide RNA. The reason being that naturally there are two guide RNAs that operate to make it active. but experimentally it has been reduced to one RNA. This RNA not only activates CRISPR-Cas9, it directs Cas9 to the particular site in the genome by base pairing. And the reason it is very simple to design CRISPR-Cas9 for new target site is this, that I made a mistake. The reason is that the Cas9 nuclease remains constant, no change there. The guide RNA, this portion remains constant all the time. Only this 20 nucleotides have to be designed according to site selected. So experimented with knowledge of DNA RNA pairing and computer knowledge, little computer knowledge can design new CRISPR-Cas9 for its selected target. How genetic changes are induced, I'll try to explain here. The nuclease produces a cut. This cut, most of the time, will be joined. The two ends of the cut will be joined. During this joining process, either something is lost or something added. Some bases are added or some bases are lost, leading to what is technically called indel production. And as a result of this loss or addition, the site of cut is modified, and consequently the gene, at least 67% of the times, will be non-functional. So any gene can then be, majority of the times, inactivated because of nuclease action. The experimenter, if he wants a particular change to be done at the cut site, then he has to provide a DNA sequence, what is referred to as donor DNA. This sequence has the change that you want to be copied, and on its two sides are the sequences that are present on the two sides of the cut. Therefore, there is pairing between the donor DNA sequence and the genomic DNA sequence. There is homology-based DNA repair. Consequently, in simple terms, the donor DNA sequence is copied into the genomic sequence, and there is sequence replacement, sequence change. And if this sequence that you have included in between contained complete gene construct, then the complete gene will be integrated at that site, and that is how transgene is integrated. So the genetic changes that are produced by gene engineering, gene editing are insertion or deletion of a few bases that leads to gene inactivation. Even whole gene can be deleted, or a selected genomic segment can be deleted. There can be sequence replacement, and this sequence replacement can correct a mutation to the wild type allele, or it can induce a desired mutation in the selected gene, and also there can be, as I said, transgene insertion. This point I made earlier, I'll just say that again. Most of the bricks will be repaired by end joining, and in these events there will be gene inactivation. This can be useful in many plant breeding situations, like tomato has a gene J2 that produces a joint in the pedicel, the stalk that attaches flower to the fruit. Consequently, when the fruit is picked, It comes off many times with a small part of the stalk and the calyx attached to the fruit. When J2 gene was deleted, there is no joint in the pedestal, so when the fruit is harvested, it comes off clean. In mechanized picking of tomato, that's a great advantage. Another example is that the first slow ripening tomato was produced by GM technology. It never reached the market, not due to lack of approval for various other reasons, intellectual property right reasons. There is a spontaneous mutation of tomato, ring locus mutation, that also inhibits ripening, but there are two features that are not desirable: indeterminate inflorescence and long calyx, long sepals. The ring locus has two genes, Limax ring and Limax MC. This is the region that is involved in determinate inflorescence and large sepals. This is the one that is involved in fruit ripening. Gene editing was used to inactivate this gene, and the resulting gene edited mutant is slow ripening, and it doesn't have large petals or indeterminate inflorescence. Anyway. This one here is normal tomato, fully ripe. This is slow ripening mutant, spontaneous mutant, and this is the gene edited one, also slow ripening. The many pathogens use host genes or host proteins to their advantage to establish infection and disease development. Blast fungus uses on-rise gene ERF922, and this is necessary for blast to infect. What was done was gene editing was used to inactivate this gene, and the gene edited line shows considerably reduced infection intensity of the blast fungus. The residual infection is because of some other genes that the blast fungus can use for its infection process. One very important example is in case of wheat. Powdery mildew fungus also requires MLO gene function for establishing infection. It is not very clear so far what MLO really does for the plant, because MLO mutants are known that function normally except under certain adverse situations when they have present difficulties. Wheat has 3 genes, MLO genes, in its 3 genomes. There is no natural mutation known for powdery mildew resistance in the gene pool of wheat germplasm. And mutagenesis, it's unlikely that it will be able to target all the 3 genes simultaneously. But gene editing was used to target a sequence that is conserved, that is present in all the 3 genes. The three genes were inactivated, and they show, they show broad-based resistance to the mildew disease. Nutritional modification can also be achieved by using gene editing, inactivating key genes that are involved in production of anti-nutritional factors. Phytate is one anti-nutritional factor that in seeds stores phosphate that is used during germination. But from human point of view and from animal nutrition point of view, phytate cannot be digested either by ruminants or by us or by poultry. Not only that, phytate interferes with uptake of micronutrients like iron. Gene IPK1 IPK1 is involved in phytate product biosynthesis. This gene was inactivated by gene editing, and the maize line that was produced by gene editing has very low phytate. This line has already been approved and is in field trials in USA. Sequence replacement experiments have been done, but only few of them have reached to the approval stage. I'll take some examples to show what has been achieved. As I said, all you have to do is provide a DNA sequence in the middle of which the sequence that you want to replace, to include into the genome in place of the homologous sequence that should be present. Resistance to glyphosate is because of two mutations in exon 2 of the gene that encodes ESPS9. You can use backcross breeding to transfer this mutant gene into a new variety, but that involves time, effort, and also has the problem of background inclusion of genes. More precise change you can bring about by taking this second exon and integrating that in place of the endogenous gene intron. And this was done by homology-directed repair to produce gene-edited plants that are resistant to glyphosate. The second is production of a drought-tolerant line by modifying a gene, argosate, that is involved in ethylene response. The increased expression of this gene leads to drought tolerance. There were two things that were done, or two strategies that were used to increase the expression level. One was to put in additional GAS2 promoter in addition to the Argos 8 promoter, and this was put into the UTR region of the Argos 8 gene. The other was to replace the Argos 8 promoter by GAS2 promoter. GAS2 promoter controls constitutive expression at a higher level. And both have generated lines of maize that are higher yielding under conditions of drought. Same thing is being done here. It's a gene sequence taken from another maize inbred line that is resistant to northern leaf blight. The gene sequence responsible for resistance to the disease was used in the donor DNA to modify the concerned gene in another susceptible line, and there was sequence replacement. The gene edited line is resistant to the fungus or the blight, and this is undergoing, have been approved for commercial cultivation, subject to the approvals by FDA and all. Downside of gene editing is claimed to be clean, highly precise, and also it would appear from the descriptions free from nuisance. It's highly precise, of course, but it's neither free from nuisance, nor only confined to where you want to make change. There are low frequencies of off-target editing. Off-targets are genomic sites that are similar to the target site that you have selected. And the sequence may differ from 1 to 3 or sometimes even by 5 base pairs. But your programmable seizure does recognize that variation at genuine target site and cut there at lower frequency. Why this off-target activity occurs? One idea is that this is evolutionary footprint left onto the system that you are using. Say, for example, transcription factors are proteins that regulate expression of certain genes that are important for the organism. As you would recognize, DNA sequence continues to change. According to one estimate of the 1,000 bull genome sequence project of USA, every animal inherits up to 400 new SNPs. So the gene's base sequence of the gene is changing. If the promoter sequence of a gene that was regulated by a zinc finger protein changed, then zinc finger did not recognize it, the gene will not be expressed, and that may have lethal consequences for the organism. So it will be to the advantage of the organism that the zinc finger protein tolerated some base sequence changes, because there is no way natural process can ensure that whenever it induces a base change in the promoter of the gene that is regulated by zinc finger protein, it also makes a corresponding change in the gene that encodes zinc finger protein. So it may be a change in the zinc finger protein, or it may be a change in the gene promoter. If there was a mismatch, this mismatch tolerance is built into the system to ensure that the organism does survive, is not eliminated. Same thing applies to CRISPR. CRISPR is the basis of defense system against DNA viruses in bacteria and archaea. And in there also, defending against the virus that keeps on mutating, you got to have a system that not only recognizes the correct sequence, but also one or few changes here and there in the correct sequence. The other thing is that there is low proportion of sequence replacement as compared to deletion insertion. That's very high. The frequency of sequence replacement is very low. Scientists have been trying everything to increase the frequency of sequence replacement. So far, it has not been successful. Nothing worthwhile. The other difficulty is that there are changes that you didn't want to happen. Say, for example, there may be a second copy of donor DNA and the donor DNA plasmid incorporated at the target site, so that you have one sequence replacement plus one additional sequence added to this. These problems, they have been greatest hindrances in arguing for using CRISPR for human genome editing. This is my first attempt at 3D painting brush, so please excuse me if the drawing is not that good. I tried to bring out the information, and I think I can convey that. This is the Cas9 protein. This is the DNA target. The Cas9 protein is mutated. Normally it would produce a cut here and a cut here. It is mutated now so that it only produces cut in one strand. This is called Cas-nicase. And then you are using a guide RNA that not only pairs with this, but it has extended portion. And this extended portion serves as the donor DNA. It carries the sequence that you want to be copied into the DNA target. That is why its name is prime extended guide RNA. So, and this enzyme here is reverse transcriptase so that it can convert this RNA sequence into DNA copy. This gene editing procedure has been termed as prime editing. It came in February, it was published in February 19, and it's being hyped as the technology that will be safe enough for gene editing in human beings. How? Since there is only a nick, not a cut, the addition, deletion of bases will not take place. Whatever happens will be by homologous recombination like situation. Since there is no donor DNA, there can be no integration of additional sequences that you didn't want. And RNA will be copied as DNA, so sequence replacement will take place. Then the RNA and the Cas9 and the reverse transcriptase, they will be recycled by the cellular proteasomal degradation as well as RNA degradation. So it should be the author's claim that it's far cleaner technology than normal Cas9 based technology, but we still have to wait what comes out subsequently. Same thing. Then there is another approach of gene editing that does not use any nuclease or DNA cutting. It only uses an oligonucleotide sequence that is referred to as chimeric oligonucleotide. This 25 nucleotide long sequence has 10 RNA nucleotides, flanking 5 nucleotides here. This is DNA nucleotides here, and there is the change that you want to bring in included in those 5 nucleotides. And this whole sequence from here to here is homologous to the genomic sequence that you want to target. What will happen that once inside the nucleus, the chimeric oligonucleotide will unfold, become linear, seek out the genomic sequence to which it is homologous and will pair. And this pairing will produce a DNA base mismatch. And this mismatch pair will be recognized by DNA repair machinery, and there will be excision repair. Half of the time on average, this will be used to repair portion will be excised and repairing will be done of this. You are not interested in this. Half of the time, this portion will be excised and repaired using this as template, and that is when the mutation takes place. In this case, there is very little risk of off-target action, very little risk of anything unwanted being happening. Only the sequence replacement will be there. The changes that you can induce, they become limited. You can replace up to 3 nucleotides. One is the most common, 2 or 3 declining frequency. Then these sequence changes can either correct mutation due to 1 or 2 base pairs or induce a mutation. They can also create a premature termination codon so that the gene can still be inactivated. And there can be also insertions and deletions produced. This will be deliberately done by the experimenter by including either additional nucleotides for insertion or deleting some nucleotides from those 5, keeping shorter sequence to induce deletion. There is at least one example that has reached already market. This is sulfonylurea resistant canola. This targets again acid. Sorry. Acetolactate synthase gene is targeted by oligonucleotide for mutagenesis. The gene was mutagenized. The mutant enzyme is no longer sensitive to sulfonylurea, and the resistant line has been released as sulfonylurea canola for cultivation both in Canada and urea. Even there, those are the countries that are proactive, as I'll note later on, but even there it took 6 years for release of this variety. You can use CRISPR-Cas9 system for either suppression of gene or inactivation of or activation of gene expression. This you do by linking to the Cas9 protein enzymes that modify DNA, either cause demethylation or methylation, or those that cause histone modification, either by causing acetylation or deacetylation, and other changes in the histone. The Cas9 is mutated to create a dead Cas9 because it is no longer able to cut DNA. It can only bind DNA, the targeted region. So you can target these enzymes to the promoters of the genes there, and please let me know 15 minutes before the time goes. I have time. Okay. You can create reagents that will seek out sites and make changes in a single base in the entire genome. Seeking out system is the same, Cas9 and guide RNA, and the base editing machinery is provided by deaminating enzymes like deaminase enzymes. Like in this case, you have cytosine deaminase. It will deaminate cytosine and convert GC base pair into AT base pair. The conversion of C to T is like this. Deamination will produce uridine. Normally, uridine will be excised and replaced by C, by the cellular machinery. Therefore, you have to deliberately inhibit this excision process of U by using uridine glycosylase inhibitor, UGI protein, and then at DNA replication time, you will be changed with T, leading to AT substitution. There has been, again, acetolactate enzyme mutated by converting a C into T at a strategic point to produce herbicide-resistant mutant. You can also carry out conversion of A to G. The important thing that I would like to point out is, at least for students, that the cell does not have DNA adenosine deaminase. It only has tRNA, adenosine deaminase, and that too in prokaryotes, in E. coli. The tRNA adenosine deaminase was taken through protein engineering process, 7 generations of protein engineering, to evolve an adenosine deaminase that acts on DNA. And this D emanates A to I for all practical purposes read as G by replication and transcription and translation machinery. But at next replication, it is converted to G. Since you can change C to T and A to G by complementary base editing, you can convert G to A as well as T to C, because you can't work on T and G for editing, so you work on the complementary strand on the complementary base, and you can make all kinds of code transitions. Base editing can achieve either it can correct a mutation due to one base, it can induce a mutation due to one base, or it can create a nonsense codon, premature termination codon, to inactivate the gene. Limitations, again the same, the frequency of editing is high, but there are low frequency for editing of other Cs in the neighborhood. There is no single base editor so far. It will edit the C, and if there is neighboring C, it will also affect that. And there are off-target site actions, and it also, in many cases, the base editor induces other changes, including deletions. Now, how that happens is not really known. This is one of the simplest devices, but very devastating device, the gene drive. In essence, gene drive is a donor DNA, a specialized donor DNA. You see, in its structure, you have a sequence here on this side that is homologous to the site of the genomic sequence where you want to put in the gene drive. Okay? But what is special about it, it has gene encoding the Cas9, gene coding the gRNA, a guide RNA, and this whole thing makes up gene drive. You can, if you want to, include a gene that you want to transfer into the population. Gene drives act strictly in crossbreeding populations. They cannot act in self-pollinated populations or in asexually propagated populations. So what will happen in gene drive is this, that if there is a gene drive individual, it mates with another wild type individual, it is heterozygous. The wild type chromosome will have target site for this guide RNA. Cas9 guide RNA will cut it here, and when repair takes place, the gene drive will be copied into it. So in the heterozygote, the gene drive is copied into the wild type chromosome to convert into homozygote. The consequence is that every generation, the gene drive frequency increases in the population. This is one pictorial representation. For those of you who are familiar with population genetics, it may be much easier to comprehend. In a random mating population, the gene frequency and genotype frequency follows Hardy-Weinberg law. The three genotypes of a gene will be P squared equal to Q squared, and that is how they will remain generation after generation, so that the gene frequency of allele A remains Q over generations unless evolutionary forces change it. In case of gene drive, what happens? This heterozygote becomes homozygote gene drive individual, and therefore the frequency of A gamete from Q is reduced to Q squared. And in every generation this will go on happening, so that in second generation it's Q to the power 4, power 16, and power 256. Since the value of Q is less than 1, the value of Q will go on decreasing. It's estimated that even if you have 99.9% wild type individuals and 0.1% gene drive individuals, in 13 to 14 generations, the whole population will have the gene drive. What it can do is this, it can modify the genetic makeup of the population so that the gene that you included along with gene drive spreads into the whole population. This can be done to introduce a transgene into open pollinated variety systems like these. You can add a gene, payload gene that is beneficial to the species. If you do that, then you can rescue a population that is otherwise threatened, and say, for example, pollinator insect species that can be rescued back, population increase can be there. There can be population elimination, and this could be done to problem-causing weeds, pests. So just like mosquitoes, malarial parasite has been one of the prime targets of discussion. It works well in the laboratory. No one knows what it will do in the field simply because there has been no field test. And the field tests are not possible unless guidelines are there, and guidelines cannot be there unless field test data are there. So you have a catch-22 situation, whether it is the chicken or the egg. Now, no one knows who will decide which. Already, but there are always enthusiasts of new technology, and they will end up doing certain things that normally, what is said, where devils dare, some people dare. In this case, there is a British startup firm that is testing a gene drive to control or eliminate Aedes mosquito, that is malaria parasite vector. Just like as CRISPR babies that were produced in China a couple of years ago. To conclude then, gene editing can add or delete two bases. It can delete complete gene. These will inactivate the gene. There can be sequence replacement, so a mutation can be corrected to normal, or a mutation can be induced. There can be also insertion of complete transgene, and this should somehow mitigate the safety concerns about transgenes. Whether it will, that is a question to be seen. To do gene drive experiments, you need certain prerequisites. Of course, there has to be a reference genome, a good quality reference genome. But more than that, one has to understand the if you want to modify a characteristic, you must understand the genes involved in the modification of that characteristic, the roles played by the genes that are involved, and the critical gene that you can tinker with to modify the phenotype in the way that you wish. And within that gene, the target site is to be selected. And once you have done this, then you can design the programmable seizure, which with CRISPR-Cas9 is much easier than with anything else. And for this simplicity, and this system, as I should mention, is not only simple to design, it is also simple to implement and very cheap. According to one estimate, I don't know how reliable this would be, to produce a transgenic event would cost something like $2,500. That was the estimate. Whereas to produce a gene-edited line with CRISPR would cost you only $35. So that's the cost difference that is involved. Now, coming to the regulation of the gene editing products, it has been said that products of gene editing should be very easily classified as non-regulated. For some products, yes, where you are only deleting the gene or inserting, deleting the gene or inserting few bases or built-in few bases, this should be treated as non-regulated. When you come to the second category of changes that you make, gene replacement, it is a messy situation, not easy to decide. What many think, and I also agree with that thought, that there will be two kinds of situations. One, where you are changing the sequence either to the wild type allele or to an allele that exists in the population and is known to occur in substantial frequencies. If that kind of change has been made, then it should be treated as non-regulated. If you are making a change that is creating a new allele, then there should be some degree of regulation. How much, that is to be decided by a panel of experts along with the policymakers and other stakeholders. Obviously, you all would agree that if you have inserted transgene by this technology, then that should undergo regular transgenic evaluation process. There are, if you look at the world scenario of regulation of new technology products, GMO and gene editing in our concerned case, the regulatory procedures, regulatory regimes that are there, they can be classified into two groups. One is product-based, the other is process-based. India follows the process-based regulation. There are a group of countries led by USA that follow product-based. Along with this product-based and process-based, the risk assessment is very important. US, this first group of countries, they follow what is permissive risk assessment. It is assumed that product of a new technology is not dangerous till it is shown there is a reason to think that it is dangerous. The precautionary risk assessment begins with the assumption that any new product is dangerous till you prove that it is not dangerous. And the consequences of, the consequences of these procedures is that in this group of countries there have been many releases of modified varieties. Only one in EU, that was a maize, BT, and only one in India, that was BT cotton, and there have been no releases. The regulatory procedures treat this group non-regulated. As I mentioned, these countries treat the wild type change and known allele change as non-regulated, others as regulated. In these countries, all are treated as regulated. The problem that arises in India, so far as I have been able to understand, is the definition in this rule, 1989 rule, that we have adopted the definition of genetic engineering that includes formation of new gene combination of genetic material by deletion and removal of parts of heritable material. This definition puts even gene inactivation process under regulation. So some change in the regulations have to be done in this country to permit gene editing to go on. If actually this does not change, then there will be a sort of stifling effect on research. It has almost stifled GM technology. It may stifle gene editing technology also. Before I say thank you, dhanyawad, I should present two slides that should be of interest to most of us, or almost everybody of us, because they concern our health. These are gene therapies that have been based on gene editing. This is a small sample of many therapies that have been approved for clinical use in universities. And they are targeting diseases that are either terminal or incurable. The chances are, in each case, the success rate varies, but the chances are very high, better than 70%, that the individual suffering from the disease will be cured. But the only problem is that the cost is prohibitive for most of us. And this Zolgensma at $2.1 million US dollars for a full course of one dose is possibly the costliest drug on earth. But it treats a disease, spinal muscular atrophy, that is definitely lethal within 5 years. So it offers a hope. More in the realm of future, this possibility I'm sharing with you. Organ transplants are a very big issue, and those who have either relative or themselves organ failure situation know how difficult it is to arrange organ donors. The technology is just beginning to emerge that may provide animals that will not provide transplantable organs, but they will provide human organs for transplant to humans. In simple terms, the technology is this, that the basic principle is that each organ it is initiated in the embryonic stage by activity of one or few genes. So one could produce an animal embryo that has that particular gene inactivated, so that the embryo by itself will not develop that organ. Say, for example, in this case, there is a PDX1 gene in mouse that controls pancreas development. Inactivate that, the mouse will have no pancreas. In this blastocyst embryo, you introduce human-induced pluripotent stem cells. These cells can be taken from the patient's dermal skin tissue, and that can be cultured and converted into pluripotent stem cell by expressing four stem cell factors. And then it can be transplanted into this embryo so that you will get chimeric blastocyst. Since the space of that particular organ will be empty in the blastocyst, the human cell will migrate into that space. And as a consequence, the chimeric animal that is produced will be animal with human pancreas. This has been shown to work with rat and mice. Inactivate mice pancreas, rat cell transfer, it is mouse with rat pancreas. But there are two questions that one doesn't know what will happen if this happens, and what is the chance of this happening. What if the human cell migrates into the brain area of the developing embryo, or worse still, into the gonad area of the animal that develops? What kind of chimeric animal we are going to get? Or what if this is used as pretext for human cloning? That's my thought. With these thoughts, I would like to finish my talk I will say thank you, Dhanyawad. But before that, an acknowledgement and appreciation and a thank in advance to two of very active colleagues in plant breeding arena, Dr. Sharan Angadi and Dr. Aparna Tiwari. They have agreed to my request for writing a chapter on commercial plant breeding. Of course, this chapter will have their subtitle, their name and everything on that chapter, to be included in the book Plant Breeding that is used by students for their study material. My idea is that if this chapter goes in there, even if it is not in the syllabus, the students might read it out of curiosity and become little exposed to the activities of commercial plant breeding. And then that way they might be better recruiting material for the companies that have them. So I want to thank them in advance that they have agreed this, and many students will join me in thanking them in advance in the hope that it is there. So thank you all very much.
Thank you very much, sir. Very masterly presentation in a textbook fashion. It has beautifully described the surgical, what are its problems, what are its prospects. Now let's see if our audience has any questions. Yes, sir.
Thank you, Professor Singh. As usual, wonderful exposition of the topic. I just want to share some information and a thought and get your reaction to it. Wonderful that you brought up this issue of gene drives, and there was this nature issue, I believe, in July, which talks about the debate on whether gene drives, particularly for tackling menaces like mosquitoes, which transmit dengue and various other things. And I believe the latest information is that in Burkina Faso, I think a group of people, I think just last month, they have released a small sample size of mosquitoes containing certain transgenes. It's not very sure whether it's a gene drive-based approach or just a transgene which actually, when they cross, it causes sterility. So some beginnings are being made in people, if you want to call it, taking some risk to see if the benefits are better than the risk. That's one. The second aspect, I was always fascinated by this gene drive system, and then thinking about how they can be used in agriculture. I think we have many problems in agriculture. Weeds is some of them. And since you touched upon the health aspects also towards the end, there is one weed, Parthenium, which we sometimes refer to as the Congress grass. Not trying to be political here, but I think everybody will be agreeing that we need to make our agricultural fields Congress book. Not to, but the point I want to make is, and that's a very high reproductive capacity kind of grass, and it can multiply millions of times. A system like using a gene drive to induce some form of sterility or lethality in the population could be thought about. And by the time we sort out these issues about regulation and whether it should be considered as non-regulated or regulated, we will at least have some technologies ready. If it doesn't go beyond the laboratory confines, we can at least add to some knowledge and understanding of the whole system in this country.
This Congress grass, the word Congress has nothing to do with India, it has to do with US Congress.
Sure.
So, when you talk of Congress Mukh, then it is not Indian Congress Mukh, it's US Congress Mukh.
It's Trump book, I believe.
Yes. The thought of weed control by using gene drive has been discussed to a small extent. One of the issues will be that the individuals that carry the gene drive, they will be weak in reproduction and then eliminated. Okay, so the gene drive will not spread unless one could have think of a gene drive that first spread and then became lethal. If that could be thought, then that would be okay. Otherwise, one suggestion was that you keep two areas. In one area, you breed gene drive individuals and then inoculate them into new area. Now that doesn't seem to be very practical to me, but I don't know. Somebody might come out with some better suggestion. But yes, please.
No, yeah, exactly. That's what I had in mind. No, not gene drive, which expresses constitutionally, but which can be triggered using an external stimuli.
I think that will be okay.
That was what I had.
So you carry a sterility gene in the gene drive, let it spread, and that is driven by, say, simply just for thought, a hormone-inducible promoter. Fine, yeah.
That was it.
To me, it seems plausible.
Well, I guess one good long question is good enough. So with this, we will come to an end. I'll hand over the mic to my chairperson, Dr. Viraktimat, to do the concluding issues.
Thank you, Dr. Anwar. Thank you, Dr. Saab. Very. The way Dr. Singh is writing books, I was about to tell him, I can't write a book on the editing. Before I said that, he told me that book is coming in the next June itself. I think it's a great news for all of us. We look forward to follow that book, sir. With this, thank you very much for a nice lecture.
Sir? Now I